And so some issues I want to touch upon are related to specifically to peak calling for histone modifications, as well as multiple hypothesis testing. And so when you actually run software to call ChIP-seq peaks, this is an example of the kind of output that you would get. So Max2 is one of the kind of super popular standard ChIP-seq peak calling pipelines or software. And so here I've highlighted in red 2 different columns. The column on the left is related to, in some sense, the signal that we see at a given peak, where the signal is basically proportional to the number of reads that map to that particular peak. And in green, I'm showing you the fold change or fold enrichment. And so that's basically, in some sense, the amount of signal you saw in your ChIP-seq assay divided by the amount of signal you saw in your control experiment. And so the larger the fold change, or the larger the fold enrichment is, basically that means that the larger the relatively larger the signal was in your ChIP-seq assay versus your control. And so a common question is, when you run one of these peak callers on your histone modification data, the 2 most important questions you tend to want to ask are, number 1, where are the most likely peaks indicated by Max2? And number 2, which of these peaks are actually reproducible? So if you run like multiple biological replicates, how do I find the reproducible peaks? Because those are going to be the ones that you tend to believe more. And so in order to answer the question of, given a bunch of peaks across the genome, which ones are real, we have to discuss the concept of hypothesis testing, or essentially go back to the discussion about p-values. And so to motivate the discussion about hypothesis testing, suppose I want to perform an experiment in which I want to test the hypothesis that I am no taller than UC Davis undergrad students. Right, so my null hypothesis is that I'm no taller than UC Davis undergrad students, and my alternative hypothesis would be I'm taller than your average UC Davis undergrad student. And so suppose in order to test this hypothesis, I perform an experiment where I say walk into a classroom or a virtual classroom, and I look at all of the students, I measure everyone's height. And so to get some terminology down, basically the height of a given student or individual would be called the test statistic. And suppose I just take a look at the room and I just calculate the number of the fraction of students who are at least as tall as I am. And that fraction would essentially be my p-value. And so if this p-value is less than 0.05, then I could basically reject my null hypothesis and say, yes, I am potentially taller than UC Davis undergraduate students. And so in the context of this experiment in which I, perform a single experiment, get a p-value, and then, either reject the null hypothesis if p is less than 0.05, or I don't reject the null hypothesis if p is greater than 0.05, there's basically 2 types of errors that can arise in this kind of experiment. And so the first type of error is called a false positive, or what's also known as a type 1 error, which basically happens when basically p is less than 0.05, and so I reject the null hypothesis. But in reality, I'm actually no taller than your average undergraduate student. Similarly, a type 2 error is what's also known as a false negative. And so that's the case where my p is greater than or equal to 0.05, and so I don't reject the null hypothesis. But actually, I am taller than your average UC Davis undergraduate student. And so both of these errors can occur due to noise or poor statistical testing or things like this. And so the multiple hypothesis testing problem can basically be illustrated as follows. So suppose that I walked into our class and I checked how many students are at least as tall as me, and I found that 50% of the students are taller than I am. And so suppose that would give me a p-value of 0.05. And then, suppose I that actually happened, and then I said to myself, okay, well, that's kind of depressing that I'm not taller than your average UC Davis undergraduate student. So why don't I just keep doing this experiment until I find a class that, in which I am taller than your average undergraduate student. So suppose I was somehow able to, jump in a time machine and walk into every classroom ever taught in UC Davis over the last 100 years. And in every single classroom, I looked at the students and I measured, everyone's height, and I checked what fraction of students were at least as tall as I was. And suppose I could do that 10,000 times. And so in theory, I would get 10,000 p-values from checking 10,000 different classes. So suppose then out of the 10,000 classes, suppose I saw one where my p-value was like 0.01. Is it right for me to conclude that I am significantly taller than UC Davis students? So even intuitively, without kind of understanding statistics, you should, when you look at this slide, you should conclude yourself, well, no, you can't really conclude that because you tested it the first time and your null, you couldn't reject your null hypothesis. And then you basically just kept searching other classrooms until you could find one where you might have just lucked out being taller than your average undergraduate student. And then, just concluded, oh, P is less than 0.05. Great. And so that is essentially the multiple hypothesis testing problem, which is that if you keep performing tests over and over and over again, you can't just look at the p-values individually and just wait till you see one that's less than 0.05 and then say, yay, I reject my null hypothesis. You have to take into consideration the fact that you're doing multiple tests. And so again, the reason why this is relevant to ChIP-seq peak calling and pretty much all other statistical tests you would do on genomics data is that on the previous slide, when I was showing you the output of the max2 peak caller, there are p-values associated with each peak, which tells you essentially the chance that you would see a peak in your ChIP-seq assay by chance. And so max2 gives you p-values for all the different peaks it calls across the genome. And so you have to, for you to, flip through this list of peaks and say, okay, which ones are real, which ones aren't, you essentially have to look, in part at the p-values and then make a judge, okay, which ones are less than 0.05, but then also take into account the fact that you're looking at multiple peaks. And so you're essentially doing multiple hypothesis tests. And so in terms of, performing multiple tests, suppose that, going back to this experiment where I walk into different UC Davis classrooms, suppose that we label each of the tests that I do, each of the hypotheses I do on each individual classroom is H1, H2 through to HM, where HM is basically the hypothesis that I was, taller than the average undergrad student in the mth classroom, right? And so essentially, if you knew the ground truth, whether or not I was actually taller than the average student or not in each one of those experiments, you can essentially build a 2x2 table, right? Where essentially the rows, basically each of those M hypotheses you test can fall into either P is greater than 0.05 for that particular test, or P is less than 0.05 for that particular test, right? And similarly, you can divide each of your hypotheses into, based on the ground truth, yes, you were taller than your average undergrad student, in which case the alternative hypothesis is true, or your null was actually true, which means that actually you weren't I wasn't taller than your average undergrad student. And so this 2x2 table essentially sums up all the possibilities in terms of whether or not you rejected your null hypothesis and whether or not your null hypothesis was actually true. And so basically the entry labeled U in this table represents the number of hypotheses in which my p-value told me, you're not taller than your average undergrad, and that was actually true. S represents the cases where the p-value actually did tell me, yes, you are taller than, or, the p-value told me that I could reject the null hypothesis that I was not taller. And it was actually true that for that population of students, I was actually taller than them. So U and S, so when U and S are large, then that's good, because that means that that my tests were accurate in some sense. I rejected the tests I should have rejected, and I did not reject the tests that I shouldn't have rejected. On the other hand, V basically refers to your type 1 errors, your false positives, because those are the subset of classrooms where I said that, yes, my P is less than 0.05, so I am taller than your average undergrad student, but that actually wasn't true. Similarly, T is your false negatives, where essentially you concluded that, no, I was not taller than your average undergrad student, but actually I was. And so something important to note in this table is that when you add up U plus T, and when you add U plus T together and V plus S together, all of those sum up to the total number of tests that you did. And so that's essentially why this is called a contingency table. And so when you talk to people and read the literature about correcting for multiple hypothesis testing, essentially what people mean, and similarly when you hear people talk about adjusting p-values for the number of hypothesis tests performed, essentially what they're usually talking about is controlling the type 1 error rate. And so remember how I said, if I was not taller than my average MCB 182 student population this year, I could just go back to previous years and keep looking until I found one. Basically, the way that you would adjust for the fact that I'm looking at multiple classes is that I would basically take my original p-values, or what's called the nominal p-value, and I would just kind of modify them such that effectively they would increase in value with the more tests I do. So the more classrooms I visit, the higher the bar should be for p to be less than 0.05. And so this is essentially controlling for type 1 error rate, in that I'm trying to adjust the p-values so that I don't call too many, I don't reject the null hypothesis too many times as I'm checking multiple classrooms. And so there's basically two really common ways of correcting for multiple hypothesis testing. The first is what's called the Bonferroni correction. And so this essentially is what's called correction for the family-wise error rate. And so what you're trying to prevent is you're trying to minimize the chances that you rejected any null hypothesis which should have actually not been rejected. And so this basically, you're minimizing the probability that V is at least 1. Right? So this is a really stringent multiple hypothesis correction, because what that means is you're trying to change your p-values in a way such that anytime you do reject the null hypothesis, it's likely to actually be that you're taller than your average UC Davis undergrad student. And so because the Bonferroni correction method tends to be conservative, and then what that means is that you'll tend to have false negatives, right? So you'll tend to not reject the null hypothesis when you actually were taller than your average UC Davis undergrad student. So people have also, it's also pretty common to use what's called the false discovery rate. And so what that does is, instead of minimizing the chances that you call any false positive, what you're doing is you're essentially controlling the expected rate of false positives. And so those produce what are called Q values. And so the idea is that if you have a Q value, so Q values are computed from P values, and their Q values are essentially what you could think of as corrected P values. And so when you report a Q value that's less than 0.05 and you reject the null hypothesis, what you're essentially saying is, well, of all the tests that I do, if I reject null hypotheses where Q was less than 0.05, then I expect that basically 5% of the null hypotheses that I rejected were actually true. And so this is basically another way of saying, well, I'm okay with having false positive rejections because I want to make sure that I don't have too many false negatives. And so in the context of peak calling, if you take p-values produced by max2 and you convert them to q values and you basically keep all the peaks where Q is less than 0.05, then what that essentially means is that you expect that about 5% of your peaks were actually false positives. And so again, as I just mentioned, some of the disadvantages of controlling family-wise error rate, or in other words, using Bonferroni correction, is that basically your corrected p-values depend on the number of tests you do. So the more tests you perform, or the more classrooms you check, then the harder it is for any given p-value to actually meet your post-correction threshold. And so you tend to have a lot of false negative. In terms of ChIP-seq peaks, if you use Bonferroni correction, you'll end up missing a lot of real peaks. Whereas when you use false discovery rate, essentially what you're trading off is you are willing to accept that some of your peaks are actually false positives, but in return, you have fewer false negatives. And so that's typically what people use. People typically use the false discovery rate.